Assalamu alaikum, students. Today we are going to discuss comparison between mitosis and meiosis. Before start of the lecture, I would like to share a word of wisdom, and it's a famous saying by an American statesman, Martin Luther King, who said, "Life's most persistent, intelligent question is, what are you doing for others?" Well, student, what do you think this note of him contains? Well, student, actually this saying actually is containing a question mark to our life. A question mark that how our life is basically spent, how our life is basically helping our fellow beings, how our life is basically supporting the one who is sitting next to you, next to oneself. It is saying that life should be lived in such a way that it absorbs the pain and suffering of fellow human being. It contains the message of humanity, the love of fellow human beings, and the care and the support and the soft corner for each and every human being. So, students, we have to spend our life. We have to spend our life in such a way that we support humanity. We support the one which is passing through misery. We should ease up the difficulties of our fellow beings. And that is also the objective as given by Allah. So please, again requesting, build up your life that becomes a blessing for others, and eventually the life will become blessing for you. Your own life will become a blessing for you. So now coming to the topic, please. The topic is actually, we are basically comparing two entities. Well, the two entities are mitosis and meiosis, right? Well, when you are comparing two entities, there should be a differential line. And if there is no differential line, then certainly marks are deducted due to this deficiency. So be careful to draw the differential line. Now, mitosis and meiosis are two types of cell division, as you know. These are typical types of cell division of eukaryotes, certainly of plants and animals. Well, mitosis is also known as equational division, and meiosis is also known as reductional division. Well, the reason behind is that in mitosis, actually, it is connected with something that is equal. It is connected with something that is equal, actually. So mitosis is the division of cell type in which the number of chromosomes actually remains the same or remains equal to that of the parent cell. On the other hand, meiosis is connected with something that is reducing or is reductional. So actually, meiosis is the division in which the number of chromosomes of the daughter cells are reduced as compared to the parent cell. And that both this description actually is itself the definition. It is the definition of mitosis as a cell division in which the cell actually divides into two daughter cells with the same number of chromosomes as of parent cell. Well, on the other hand, meiosis is the division of eukaryotic cell of a eukaryotic cell that is 2n or diploid in nature, and it divides into four daughter cells that are haploid, means they consist of half the number of chromosomes as compared to the parent cell. So this was the first difference, and now we will move on to the next one. Well, the next difference is discovery. Well, you can write, technically it should be right, it can be written in the first part, but preferably the definition should be placed in the first place as per norm of our biology and science. So discovery, actually mitosis is and meiosis, both of them are their discoveries, and their description is contributed by German scientists, German biologists, and really students, we should salute the Germans as their contribution to building up the cell theory, and the first statements that were of Schwann and Schleiden, you remember. So hats off to the Germans, and mitosis actually was discovered by Walter Fleming in 1880. And on the other hand, Oscar Hertwig actually discovered and described certainly meiosis in 1876. Other thing is, apart from it, occurrence. Well, this mitosis, mind it, occur in each and every body cell of you except the reproductive cells, right? And on the other hand, this meiosis actually occur in only the reproductive cells or germ cells or gametes, but not in other germinal body cells. So this is the basic difference of occurrence. It occurs only in somatic cells, germinal body cells, throughout the body, and on the other hand, it occurs in the gametes that are the reproductive cell but not somatic cells. Well, the next thing is the number of divisions. Well, mitosis is basically a one-step division. It is technically a one-step division, and those one steps basically consist of 4 stages. On the other hand, meiosis actually is a 2-step division and containing 4 plus 4, 8 stages. The next difference is prophase and prophase 1. Well, in the prophase of mitosis, there is no pairing of homologous chromosomes. No homologous pairs are formed. On the other hand, in prophase 1, well, the homologous chromosomes are paired. And number 2 is actually the formation of synapses, tetrad, and chiasmata are not present in mitosis, and all these characters, that is the formation of synapses, tetrad formation, and chiasmata are involved in meiosis. There is no exchange of genetic material, and you know that it is called the crossing over. And in mitosis, in the case of mitosis, and in the case of meiosis, there is an exchange of your genetic material, and that is known as crossing over. So meiosis contains this process of crossing over, and due to these processes in meiosis, there is a wide range of genetic recombination. Well, a wide range of genetic recombination in the daughter cells. On the other hand, in your mitosis, there is no such a thing as we have mentioned earlier. These the above three steps, or three steps, are not occurring, and certainly there is no genetic recombination, and the daughter cells are completely similar with the parent cell. Well, students, you can see from the diagrams actually. Well, this diagram is certainly representing the prophase of mitosis, and this diagram is representing the prophase 1 of meiosis. Well, student, the thing is that you can see from the diagrams that if yes, in prophase of meiosis, spindle is formed, your centrioles are moving towards the pole, yes, your nuclear membrane is disintegrated and eventually it disappears, yes, and most of these processes, the nuclear envelope disintegration, the movement of your centrioles to the opposite poles, spindle fiber formations are same. In both the cases are same, but the difference actually is, as is evident from the diagram, you can see. Well, here you can see there is the pair. This is the a pair of homologous chromosomes, and these are pair up. You know, these are pair up. Well, yes, a group of tetrade, a group of 4 basically is made here. Yes, you can see, and there is gas meta here. Yes, the point of attachment, and it will be certainly passed through the crossing over as well. But on the other hand, if you see on the prophase, well, there isn't any pairing. Well, the sets of chromosomes are just you know, they are just moving with a distance, and with no chromosomes, no pairing up of homologous chromosomes, no chiasmata, no crossing over, and no synapses are here. So this is a major difference. Among other similarities, the major actually difference is this. And due to this difference, actually, due to this arrangement and these processes, actually, this helps in the genetic recombination. And now we will move on to the next phase. Well, student, the next phase is your metaphase. Metaphase in mitosis and metaphase 1 in meiosis. Well, metaphase is actually, as its names show, it is the one thing that you have to remember is the arrangement on metaplate. It is the arrangement of chromosomes on the metaplate. Well, in mitosis, it is different. In meiosis, it is different. In mitosis, single chromosomes are actually attached on the metaphase plate. On the other hand, in meiosis, actually homologous pairs align to form metaphase plate. So this is the technical difference between mitosis and meiosis. It is evident from the diagram. Well, you can see from here, well, single chromosomes. Well, these are the chromosomes with their sister chromatids. Well, and they are longitudinally attached one over the other and over the metaphase plate. Well, equatorial plate, one can say. And so, on the other hand, metaphase 1, you can see, well, this is the arrangement that is totally different and contradicting from this one. Totally contradicting, and certainly the contradiction is that they are attached in a pair sort of arrangement. Well, they're pair sort of arrangement of homologous. Well, these are homologous, similar sort of chromosomes. These are smaller, you can see. These are homologous chromosomes, and this here is making the metaphase placed or equatorial plate. Well, the next difference is of anaphase. Well, anaphase is the process of mitosis, and anaphase 1 is the process of meiosis, right? Well, metaphase was the actually the arrangement on the metaplate, yes, and anaphase, my dear student, is detachment. It is actually the detachment of chromosomes. Well, in anaphase simple, well, individual chromosomes are actually broken. Individual chromosomes are broken. Eventually, individual chromatids are pulled towards the opposite poles. On the other hand, individual chromosomes are pulled towards the opposite pole. Anaphase, in anaphase 1, your individual chromosomes is pulled from that pair of homologous chromosome actually, and in mitosis individual chromatids are pulled. Well, this is the difference. This is the difference which is indicated here. You can see from the diagram. You can see from the diagram here in anaphase 1. Well, you can see actually the chromosomes, the full chromosomes are basically detached and are pulled back from the pair of homologous chromosomes. Well, that tetrade set, you know, from the tetrade set, actually the chromosome here is pulled by spindle fibers here, and here a chromosome, full-fledged chromosome with two sister chromatids is pulled like here. On the other hand, the scenario of mitosis is different. Well, these are simple sister chromatids. The chromatids are pulled here. Well, the chromatids is pulled here from that set of chromosome, and its sister chromatid is pulled here, on the other here. So here individual chromatids are pulled, here individual chromosomes is pulled. Well, the next phase is your telophase. Telophase in mitosis and telophase in meiosis, telophase 1. Well, the daughter nuclei contain basically in the telophase, actually what happens is, as you have studied in the earlier lecture, telophase is actually the reversal. It is the reversal of prophase, right? And similarly here, telophase 1 is also the reversal of prophase 1. Well, the resultant actually is the difference. There are similarities certainly between them. Well, certainly the spindles are disintegrated, yes. Your centrioles basically are separated and moves to near the nucleus, and individual nuclei basically are formed. Well, your decondensation of chromosome occur, yes, these are all the similarities. But here, actually the difference is in telophase mitosis, diploid number of chromosomes are obtained. Diploid number of chromosomes are obtained. And certainly each chromosome here each chromosome consists of single chromatid. Remind it, boys. This is the technical point that should be kept in mind. Single chromatid here is making the chromosome. Well, on the other hand, telophase 1, where daughter nuclei contains haploid number of chromosomes. Well, number of chromosomes. Well, here chromosomes actually with their sister chromatids actually represents the full chromosome. Here only single chromatid represents the chromosome, but here it is not single chromatids. That is the difference. This is haploid, that is half the number of from the parent cell, and this is diploid, that is equal from the as per the parent cell. Well, you can see the diagram actually. Well, this is the diagram showing the telophase of mitosis, and certainly nucleolus is formed. Yes, your centriole is moved towards one side, and near the nucleus, nuclear envelopes start developing. Yes, and well, you can see actually that there is these are decondensed chromatids. These are decondensed chromatids, sister chromatids only, that are playing the role of chromosomes, that are chromosome. Well, on the other hand, telophase 1, you can see, my dear students, that a full-fledged chromosome with two sister chromatids basically play the role of chromosome. Here it is consisting of sister chromatids as well. Here as well. Here, well, this will be one chromosome, and this will be another chromosome, and this will be another chromosome, and they are haploid in nature, that is half the number from their parent cell, from their original parent cell. So this was a difference, a major difference, telophase and telophase 1. And now we can have a bird eye view of the whole processes of mitosis and meiosis. Well, you can see the prophase in both the cases, prophase occurring, and well, in here actually prophase, your pairing up of chromosomes and homologous chromosome, and in this case there is no pairing up. There is no pairing up, and in other words, there is gasmata is here, synapses basically occur here, tetrade here, crossing over here. All the processes basically occur here, and not in mitosis certainly. I'm talking about meiosis. And in anaphase, in anaphase actually the separation of the chromosomes basically occur, and in the previous one was the metaphase. Well, this is the metaphase, and in the metaphase the arrangement was done on the metaphase plate, and eventually telophase, and in the telophase eventually the cells are formed. In the prophase, well, the next stages are the stages of meiosis 2. Well, this is a comparative diagram which is representing the cell division. Well, now coming to the significance of mitosis and meiosis. Mitosis is significant and helpful because it helps in development and growth, cell replacement, regeneration, and asexual reproduction. On the other hand, meiosis basically helps in maintaining the number in the next generation, as well as the variation to produce variation in the next generation. Errors, faults, defaults, or problems that occur due to the mitosis is the famous problem is the formation of tumors of cancer. On the other hand, meiosis, the errors, sometimes non-disjunction in chromosomes occur. That is, this disjunction is the separation of chromosomes. In the anaphase you have observed, well, due to the non-disjunction, improper disjunction or non-disjunction, the number of chromosomes is disturbed, eventually an abnormal offspring is produced. Well, this was all about the lecture. Now brainstorming questions. What is the difference between prophase and prophase 1? Haploid number of chromosomes are obtained in which divisions, and what would happen if there is no crossing over in meiotic cell? Well, you have to do the task of differentiating mitosis and meiosis, and to draw the diagram differentiating process of mitosis and meiosis. Well, this was all about the difference and comparative analysis of both the processes, and I wish best of health, and please stay home and stay safe.